Hello, my name is Noelia Che, and I am a postdoc from UCL and a John Goldman Fellow working on acute myeloid leukemia. My research mainly focuses on one big question: how do leukemic cells survive? And even under an environment with limited nutrients and resources, oxygen, and even with the exposure to the drug, they manage to survive. So I'm very interested how. In this project, I am focusing on a subtype of leukemia, acute myeloid leukemia, also known as AML. What we know at the moment is AML cells are highly dependent on a protein called MEB to survive and for disease progression. And MEB is a master regulator that controls everything, for example, like cell proliferation and self-renewal. So you may ask, so if that is such a driver of the disease, we just get rid of it and then patients got a cure, easy peasy. But unfortunately, the answer is no. It's not that simple because it controls everything. If we just get rid of it, the patients will not have any blood cells and they would just die. So for my project, it's particularly looking at its downstream pathway that would be unique to the leukemic cells. So hopefully if we target that and then we just simply mainly specifically targeting the leukemic cells rather than normal blood cells, then the patients will have an alternative treatment. What very interesting is what I discovered is MEB is actually control the metabolism of AML cells. This is very interesting because, as we know, 100 years ago, cancer cells tend to using a different metabolism than compared to other normal cells. So what I find is MEB is actually helping the cells to adapt under the hostile environment. So this is why they are so clever. They are like having mini brains. They can, when you apply some selective pressure on it, they can just change and then I can manage to survive. And so this is what I am working on at the moment. Receiving a John Goldman Fellowship means a lot to me because it is one of the very few schemes out there that help young career researchers like me during the transition to an independent researchers. So with this invaluable opportunity, I will be able to develop my own niche in research and then also building up my foundation. Hopefully I can apply other larger grants for mainly focusing on how leukemic cells utilizing energy resources, and then we can target them uniquely in the future.